Right. Well, Richard, thank you very much for joining us this morning. The first question I'd like to ask you is, can you please tell us about your journey to Scancell?
Well, it's been a long journey. It started 20 years ago. And like so many things, it happened through a personal introduction of a friend of a friend. I'd just given up corporate life and was looking for other interesting things to do. And Lindy was just starting out on her journey and needed some advice and help. So I met her and we had a chat. And at that time, it was Dr. Lindy Durrant. She hadn't been elevated to professor. And she had been talking to a colleague in Obs and Gynae who was very interested in the development of a non-invasive test for Down syndrome. So Scancell was actually a little diagnostics company, hence the name Scancell. That's how it started out. And I met her at that time, and Lindy had found an antibody which would allow her to sort fetal cells in maternal blood. And once you'd got the fetal cells, you could do a chromosomal analysis, in theory, and see if the fetus was normal or not. And then what she was looking at was Down syndrome in particular. And that's what we did. And there were a tiny number of people, no money. That went on for a year or two. But during that period, I found out that Lindy's freezer was actually full of 30 or 40 very exciting antibodies that had the potential to treat cancer. I said, well, for goodness sake, why don't we think about making a cancer therapeutics company? And then, to cut a very long story short, the fetal cell test wasn't commercial. The cancer therapeutics were. And we started out, raised new money, and started a cancer therapeutics business. And that was back in the early 2000s. And that's how the whole thing started.
Wow. Okay. That's quite a pivot then.
Well, it was, but things are not necessary. The things don't go in a kind of linear fashion, do they? You have to be open to new ideas. I mean, that's the key. You set off on one journey. Suddenly, you see something to one side that might be interesting. Turns out to be even more interesting than the first idea, and that's the basis for the business. And in a way, that's how Scancell has developed over the last 20 years, exactly in that way.
Fantastic. Can you give the elevator pitch as to what Scancell does now?
Well, Scancell's a cancer immunotherapy company. And it's essentially developed along two lines. One is through the development of cancer vaccines, and one is through the development of monoclonal antibodies that target cancer. And the cancer vaccines, it's always been about the generation of what are called cytotoxic T cells. These are T cells that are specifically directed toward a particular cancer that are so potent that they kill the cancer cell. And that's what the cancer vaccines are intended to do. And there are two platforms. One's called ImmunoBody, which stimulates one particular arm of the T cells called CD8 T cells, and Moditope that stimulates CD4 T cells. And those cancer vaccines platforms are both in clinical development now. And alongside that is really where the whole story started for Lindy, which is monoclonal antibodies, which we've come back to. And whilst she was developing these cancer vaccines, she found a particular type of molecule on the surface of cancer cells called glycans, which are sugar-related molecules, which were uniquely associated with cancer cells but not healthy cells. And that's the holy grail of any pharmaceutical, really, to be so precisely targeted against the cell you want but not the healthy cells. And some of these antibodies target very specific molecules on cancer cells. And that's called the Genmab platform. And then alongside that, you've got the Avidimab platform, which can enhance the potency of these Genmab antibodies, and indeed any antibody. So you've got a diversified platform of 4 technologies, all within the cancer immunotherapy field.
And you may have touched on it in the previous answer, but what makes Scancell different to other biopharmaceutical companies?
Well, in a sense, it goes back to what Charles was saying at the beginning. I've worked in and for and with quite a number of companies. Started out with Astra at a time long before it was AstraZeneca. And Astra, at that time, a very Swedish-based company, which was very unlike other pharmaceutical companies in that it was very science-driven. And that science-driven research led to wonderful compounds like omeprazole, for example, which would never have emerged onto the market were it not for being a science-driven company. And one of the things that attracted me about Scancell and Lindy was that everything about the company then and now is science-driven. So it's science-driven innovation. And the way things worked over the last 20 years between me and Lindy was that she was able to continue that science-driven innovation. But of course, it has to come within a commercial framework. And that's a long-term commercial vision for the future. And I provided, in a sense, that long-term commercial vision for the future. Lindy continued to be able to follow her passion and her science. And that strategy takes a long time. And I was very conscious of Charles's comments about time. And if you start from a light bulb moment, and all Scancell's projects have started from light bulb moments, it takes a long time to develop those into products, clinical entities, and things that you can sell. And that's just beginning to happen now, to my great delight. So I would say the combination of science-driven innovation, long-term thinking, commercial orientation, and by the way, long-term supportive shareholders as well, who are prepared to have been prepared to join the company on that journey, and I'm beginning to see the payoff now, and even more in the future, I believe.
Richard, if I could just interject a question, because I know from previous conversations that in the days when you were in corporate life, you had a particular approach to commercialization which appealed to me, which is what I would summarize as being a light approach. And I just wonder whether you just give a couple of quick sentences on your approach to commercialization of a global opportunity.
Gosh, I don't quite know how to answer that question. What did you mean by a light approach?
I always felt you adopted a very strategic approach where you attempted to achieve global reach by collaborations and relationships rather than building big central operations.
Well, this is one of the most challenging things for any biotech company, I think. I mean, everybody knows that the pharmaceutical business is a global business. And nobody's going to be interested in any product that is limited to a country or a region. And every biotech company's ambition, of course, is ultimately to have a global presence and have all the benefits of the margins that selling your own products can achieve. But there's a huge journey between starting up such a company and achieving that ultimate goal, and very few companies do it. Most of them are American. There are some in Europe that are beginning to emerge after 20 years, a journey like Genmab and Argenx but they're still finding it difficult to achieve it. So the only way you can move forward is, as Charles said, through collaboration, trying not to give too much away, always having to give something away, learning from partnerships, growing gradually, all the time being aware of the desire over time to increase shareholder value. And you have to do that in stages. And those companies that can, shall we say, hold on to enough to achieve that shareholder value whilst in parallel using and leveraging the help and support of companies that are further down the road is the way to do it. It's an extremely hard balance to achieve.
Great, thanks.
I think, when combined with the recent Genmab announcement, that's a very revealing thought process.
Well, Genmab, I'm very fond of Genmab. I've known them since they were 6 people in an office in Denmark. The company in those days was run by a lady called Lisa Drakeman, who's the wife of Don Drakeman, very well-known US entrepreneur. Their finance director, Michael Jensen, their chief scientific officer at the time, Jan van der Winkel, who became chief executive officer. They're an antibody company who've evolved in exactly that way. Over time, very long-term vision. They had the good fortune of some very wealthy long-term shareholders, one of which is Red Mile, by the way, which is also
right.
Scancell's largest shareholder. And very often these connections are not entirely coincidence. And I've been talking to Genmab for 20 years on and off. And it took 20 years, pretty much, to get this very, very nice deal that the company was able to announce a short while ago around one of Scancell's preclinical antibodies, one of the Genmab antibodies that I mentioned. So it's another example of patience, good science, working with the right people. Genmab is going to be a fantastic collaborator. They have, after 20 years, only just started to build, in collaboration with Seagen, their own marketing presence in the United States. And they've got another collaboration with AbbVie, which will allow them to expand that process. But it's, this is a very long-term process.
Yeah.
Okay, so something we always like to talk about on these shows is what is the company's golden nugget? So, what's the kind of what's the rich seam of gold that they have or are going to hit? So in your opinion, what is Scancell's golden nugget?
Well, that's a tough one, really. One of the things we've tried to do is diversify the portfolio. And one of the reasons for that, especially in immunotherapy, and cancer immunotherapy in particular, it's very difficult to predict from animal studies what's going to work in the clinic. So you can get fantastic results in animals, and it's not quite as good in the clinic. You just don't know. And that's because the immune systems of mice and men are quite different. You get an indication. That's for sure. However, with all those provisos, nothing comes close to the excitement around Moditope. And one of the reasons for that is that Moditope stimulates an arm of the immune system in a way that we'd not seen before. These are what are called cytotoxic CD4 T cells. CD4 T cells are normally helper cells. It's normally the CD8 T cells that do the work. And quite unexpectedly, the Moditope platform stimulated cytotoxic CD4 T cells. And that translated in animals to the most potent anti-tumor effect that Lindy and her team had ever seen. It was quite difficult to translate that finding into a product. Manufacturing was complex. No one had tried to make these molecules before. It's been done. It's now in the clinic. And the first few patients, it seems as though we're generating an immune response as we would like in patients. And one of those patients has seen an anti-tumor response. a late-stage patient. Very early days. But if this works out, this is indeed going to be one of the most exciting transformational findings. So that is probably the golden nugget.
Mm. And just, I mean, a theme we've hit on a few times is time. So when was Moditope first conceived?
Well, it was 2011 the discovery was made, and it was a serendipity discovery. And Lindy is an intuitive scientist, and very often the greatest advances in science are serendipity. As you know, and Alexander Fleming is one of the classic ones. It takes a great scientist to do an experiment. It doesn't really achieve what the experiment set out to do, and you get that's a bit odd moment. You look at it, look at the data, that's a bit odd. And it takes that kind of leap to go to the next phase and say, I wonder why that odd finding happened. That's how it happened with Moditope. It wasn't a clever plan to discover citrullinated peptides that could stimulate CD4 T cells that might kill cancer. It was a chance finding. But you need to have an intuitive, very sharp scientist to spot that serendipity moment and take it forward. That was back in 2011. We kept it secret until 2012 because we needed to file some patents. And then we announced it in 2012. Memorably, the share price went up about 5 or 6-fold in very short order at the time.
I'd just like to talk about Lindy for a moment, if you would, Richard. So Lindy is your CEO and I guess the chief idea generator. So what makes her such an effective leader of Scancell?
Lindy is completely passionate about what she does. So I don't think there's a waking moment that she's not thinking about Scancell. She's absolutely determined to succeed. And I'll give you an example. When we used to travel to the States on business, I'd be drinking a glass of wine and relaxing ahead of the 5 intense days that we'd have meeting companies and potential investors. Lindy would use the time to read about 50 scientific papers that she'd printed out and brought with her in her briefcase.
Wow.
Whilst I was enjoying myself with the claret. So, and that's how she was. It's not to say that she doesn't relax, but I think she's totally committed and passionate about her science and is utterly driven to achieve success. And she won't give up on any idea until it's done. And I think that's one of the main reasons that the company has been successful and will continues to be so.
The final question on the schedule then is, what is your? I was just wondering if you could talk about because you're recently retired from Scancell. So I was just wondering if you could just talk a bit about your fondest memory or your fondest memories from your time there.
Well, I mean, there are many. It's been a very exciting ride and numerous stories of success and despair following one another. But there are several. I mean, one right at the beginning, which Lindy and I remember with great amusement, is when she had this fetal cell sorting technology. Back in the early days, and somehow we got the attention of Roche Diagnostics, and this is very exciting. We had no money, very few people. We'd sorted some fetal cells with this rather good antibody, and it was all looking quite interesting. So we cobbled some money together to get the cheapest flights we could to San Francisco, which was the headquarters of Roche Diagnostics. Stayed at the hotel on Fisherman's Wharf, And the next morning, asked the concierge if they could get a taxi to take us to Roche Diagnostics. 15 minutes later, to our horror, a long white stretch limousine emerged outside the hotel door. It was too late to change anything. And we were appalled. Who on earth are they going to think we are? So off we went in the stretch white limo to Roche Diagnostics. And embarrassingly, it stayed outside their front door for the hour that we were there until we were ready to go back again. And we had a very nice meeting with them, but we were very embarrassed that we turned up in the stretch white limo. And we went back to the hotel and flew home again. And we didn't get the deal at the time, but we'll never forget the stretch white limo. I mean, there were other wonderful occasions as well. I mean, we were talking about Moditope before. One of the things I will always remember was back in 2011. I'd never even heard of Moditope at this time. And when Lindy and I used to communicate by email, the most I would get from Lindy would be 2 or 3 lines. And then suddenly, over the email, I got a page and a half. It was a kind of stream of consciousness thing. I've still got the email printed out in my office, which basically was announcing the serendipity discovery of Moditope and her excitement about it, and how this was completely unexpected, and how it was going to save the world and all the rest of it. And that was how Moditope started. And the first I heard of it was this. She couldn't keep it to herself. This fantastic news. And we had to keep that a secret, as I said earlier, for a year, whilst patents were filed on the use of any citrullinated peptide for the treatment of cancer. And, the rest is history. And then, having filed those patents, we announced it, which Charles remembers, a year later. But that was another story in Scancell's history that's very precious and good fun to remember.
I can imagine. It's great receiving those kind of emails, isn't it?
Yeah, absolutely.
Could end up being a valuable museum piece one day.
Yeah, scribbled all over it, but it's the real thing.
Great. Well, I think that's everything, Richard. So that's been, for me, that's been absolutely fascinating to hear all that. So thank you very much for your time.
My pleasure.
And I think we'll leave it there. So thank you.